Hello viewers, welcome back to another video on special senses, specifically the eye. So in this video we're going to talk about the photoreceptors, which are the cones and the rods. Remember I said when I was talking about the retina and the functional anatomy of the eye in the previous videos, we said there are photoreceptors located in the retina that are involved in the transduction, the changing of light rays into action potentials that are then transmitted to the brain in order for perception of vision to occur. So these are the ones that we're going to talk about today, and we're going to specifically talk about their function as well as their anatomy, how their structure in short. So we have two photoreceptors, the rods and the cones. So these are named as the rods because of their rod-like structure. All these are conical in shape. So they have got, both of them have got three parts. The outer segment, you can also see the outer segment here, the inner segment, synaptic terminal, and synaptic terminal. Those are the things that are common to these photoreceptors. Now the outer segment for the rod, has got discs known as membranous discs. So these are just simply membrane bound, flattened membrane bound intracellular organs that detached from the membrane. So they're just intracellular organs, or I would say intracellular organelles that detached from the membrane, but they're just free floating. They are membrane bound and free floating organelles that detach from this membrane. That's what membranous discs are. Unlike here in the cone, you have what circules rather than membranous discs. These circules are just infoldings. It's just the outer membrane or the plasma membrane that is folding inwards like that to form these circules. Here they are actually intracellular organelles that detach from the membrane that form those flattened membranous discs in the outer segment there. Then the circle and the discs have photosensitive compounds in them. They have photosensitive compounds in them that get to react to the presence of light. So the photosensitive compound in the rods is the rhodopsin, while in the cones there are three. One, cyanopsin for blue color. The second one, iodopsin for green color. Then the last one, porphyropsin. Something like that, for red color. Those are the photosensitive organs that you're going to find, photosensitive compounds that you're going to find in different cones. So if the cone is for blue color, you find cyanopsin there, and so on. Then in the inner segment of both, that's where production of photosensitive photosensitive compounds is happening. The inner segment functions in production of photosensitive compounds. Then you also have mitochondria in them. Also have mitochondria in the inner segment. as well as the nuclei here, or we can say the cell body. Then we have synaptic terminals for both of them, and both of them release glutamate at the synaptic terminal. They release glutamate at the synaptic terminal. Then we're also going to talk about the functions of these. The functions of these cones and rods in the transmission of in the visual processes, in short. But before we do that, there's a part here connecting the inner and the outer segment known as the ciliary stalk. Ciliary stalk. This one is where when the photosensitive compounds have been synthesized in the inner segment here, they'll pass through this ciliary stalk, then they'll go into the outer segment there of the cones or the rods. Now, the rods actually are very sensitive to low threshold light. So they are used for scotopic or night vision. Scotopic vision or night vision. The cones are sensitive, very sensitive to high threshold light. We say light stimuli. Even here, light stimuli. Very sensitive to high threshold light stimuli for the cones, while the rods are very sensitive to low light stimuli. And the cones are used for photopic vision or daylight vision, where you have got bright light, because bright light is the stimuli for the cones. They're actually high threshold light stimuli responding photoreceptors. Then for the rods, do not participate in what? Do not participate in color vision. Do not participate in color vision, and they have low visual acuity. In the previous video I said visual acuity is a bit for the eye to determine the precise shape as well as features of an object, or when we put two objects together in at a short distance, or when we put two objects together close together, they should be able to differentiate those two. The ability to differentiate those two objects is what visual acuity is about. So the rods have got low visual acuity. They are also found more on the periphery of the retina. These ones, the cones, have got high visual acuity, as well as participate in what? Color vision. Participate in color vision there. Then there are more cones are more on the what? Are more on the fovea. So the fovea only has cones, doesn't have rods. The periphery has got both cones, but has got more. The periphery of the retina has got more of the rods there. So both release glutamate, like we have said. In this one, glutamate as the neurotransmitter there. So that is all about rods and cones. They have the outer segment and the inner segment, as well as synaptic terminal. The rods have got membranous discs, all this has got circuits. They both are the mitochondria, the cell, the cell nuclei there. As you can see in the inner segment is the one that functions in the synthesis of photosensitive compounds that are present in the outer segment. in the discs for the rods and in the circuits for the cones. And they'll say here the photosensitive organ is rhodopsin, while here you have got cyanopsin, iodopsin, and porphyropsin for blue, green, and red respectively. Then the other thing I've talked about that the rods are for scotopic vision or night vision, while the cones are for photopic vision. The rods are for low visual acuity and do not participate in color vision, while the cones have got high visual acuity and participate in the color vision. And I've also said the periphery of the retina, that's the point where I've got more rods than cones, while the cones are found more on the fovea, while the rods are also not present where on the fovea. Then they both release glutamate. So with that said, we've talked about the structure of rods and cones. As usual, don't forget to subscribe, comment, like, as well as suggest topics that you want us to do for you. Thank you very much.